But then there's evidence that counters that. For example, this. Seagoing fishermen were in the North Atlantic in the first millennium BC, and probably earlier, indicated by bones of deep sea fish in coastal sites of northeastern America. Well, how do you get deep sea fish? You have to have a boat. You have to have a way to go out and get those fish, and yet we find deep sea fish bones in the archaeological remains of some of these people in northeastern America. They go on to say that the probability of transoceanic contacts before Columbus is so high, one might say it is a statistical certainty. I concur with that very much. In fact, this image was actually on a wall in the Valley of the Kings in Egypt, and it clearly shows a boat. This far predates Lehi and his group leaving on boats. So to think that these people did not have the capability of building boats and traveling really all over the world is not a very sound idea. Especially since my personal belief is that I believe that people have known how to build boats ever since Noah built the ark. So that makes more sense to me. American Journal of Human Genetics, 2003 article. This is where it gets really more interesting. It says the results of this study point to the following conclusions. First, that haplogroup X variation is completely captured by two ancient clades. Now, haplogroup X, if you take the main haplogroup X group, it can be subdivided into two subgroups called clades. They called one X1, the other one X2. X1 is largely restricted to North and East Africa. X2, on the other hand, is spread widely throughout Western Eurasia. Where's Western Eurasia? It's essentially the Mediterranean area. So we have X2 is a Mediterranean type group. Second, it is apparent that the Native American haplogroup X mitochondrial DNAs derive or actually stem from X2, the Mediterranean group. That is very important to understand what is happening here. Third, the few Altain samples that they had from the Siberian haplogroup X lineages are not related to the Native American cluster, and they are more likely explained by recent gene flow from Europe or from Western Asia. So that they said the Altains are not related, in fact, to the Native American groups. So what we have here is this. X1 DNA, which is found primarily in North and East Africa, and X2 DNA, which is found in the Mediterranean area and in the North American area, and there's really no understanding or explanation of how it got here. Finally, phylogeography of the subclades of haplogroup X suggests that the Near East is the likely geographical source for the spread of subhaplogroup X2. The presence of a daughter clade in northern Native Americans testifies to the range of this population's expansion. It is notable that X2 includes the two complete Native American X sequences. And that is really a very powerful thing. How did X2 get from the Mediterranean area into the Americas? There's been many different theories of how it arrived here, but one that would certainly make a lot of sense is if you take this book, you see, that was found on gold plates in a hill in western New York called the Book of Mormon. That has a very plausible explanation of how this X2 may have arrived here. But let's talk a little bit in more detail and more depth on this. I wanted to find out what other populations have this haplogroup X DNA. And I found a group. It was called the Druze. Who are these Druze people? Well, according to this BMC Evolutionary Biology in 2007, there was an article that was talking about the Saudi Arabian population. In this article, it says that the Druze sample was significantly different from all other populations, mainly because of a high frequency of haplotypes that belong to the minority haplogroup X at 27%. Well, where are these Druze? What is their origins? Now, the Druze, just a quick background on the Druze, they are a religious order. They actually do not allow new members or converts to come into this religious order. The only way you can become a Druze is if someone else dies or if you're born into it. And because of that, they actually have remained quite isolated from the other populations the host populations that they live within. But here's where the Druze come from. This is from Israel Central Bureau of Statistics, 2006. The Druze reside primarily in Syria, Lebanon, and Israel, with a smaller community in Jordan. Israeli Druze are mostly in Galilee and around Haifa. The Golan Heights, a region controlled by Israel but claimed by Syria, is home to about 20,000 Druze. So here we have a population that has haplogroup X in a fairly high frequency that is the same type of X as, they're both X, haplogroup X DNA in the Druze population and also in the Native American populations over in North America. This is the worldwide distribution of haplogroup X frequencies. You see that they are primarily, the Druze population is about 26% there in the Mediterranean area. Ojibwe Indians, on the other hand, are about 25%. Then we have the Orkneys just up in Europe. So we know that it's a European lineage, and it's found in Native Americans in North America. This is new information that is from May of 2008 in PLOS 1. It's called the Druze, a population genetic refugium of the Near East. A refugium is actually typically used in ecological terms when they're talking about an environment that hasn't changed over a long period of time. And they're using that word here because they're saying that the Druze population, because of their policy of not intermarrying with other groups, have actually maintained and they've become isolated from their other host populations, and therefore they haven't changed much. The findings were enabled through the use of paternal kindred-based sampling approach and suggest that the Galilee Druze represent a population isolate, and that the combination of a high frequency and diversity of mitochondrial X haplogroup signifies a phylogenetic refugium, providing a sample snapshot of the genetic landscape of the Near East prior to the modern age. In other words, geneticists are now using the Druze population as the sample population for the Near East prior to the modern age, which I think is very significant since they have this haplogroup X that matches up with, or that links them with potentially links them over with the Native Americans in North America. This is talking about Jewish and Middle East non-Jewish populations. This is showing how the Druze population is closely related to the Jewish population from a genetic standpoint. The Ashkenazi, Roman, North African, Near Eastern, Kurdish, and Yemenite Jewish populations formed a fairly compact cluster between the North African and European Jews. This Jewish cluster was interspersed with the Palestinian and Syrian populations, whereas the other Middle Eastern non-Jewish populations, meaning the Saudi Arabians, the Lebanese, and the Druze, closely surrounded it. So they were very close to the cluster of the Jewish populations. The close genetic affinity of Jewish and Middle Eastern non-Jewish populations was confirmed in population differentiation tests, which means that the Druze population is quite closely related to Jewish populations, but it isn't a per se Jewish population. They are actually more closely related to the Lebanese and the Palestinians than they are Jewish populations. But they still have this haplogroup X. This is from Epic to Canon, History and Literature in Ancient Israel, by Frank Moore Cross, 1998. The social organization of the West Semitic tribal groups was grounded in kinship. Kinship relations defined the rights and obligations, the duties, status, and privileges of tribal members. Kinship was conceived in terms of one blood flowing through the veins of the kinship group. Kindred was of one flesh, one bone. What he's saying here is that Western Semitic groups, like the House of Israel, were grounded in kinship, that they didn't intermarry with other groups, that they maintained a relationship among their people, among their lineage. This is from 2008, called Counting the Founders, the Matrilineal Genetic Ancestry of the Jewish Diaspora. Hence, the lack of these particular chromosomes among North